Hey guys, and today I'm going to be teaching about transcription, translation, and mutations. One example is the famous cartoon by Nickelodeon, the Teenage Mutant Ninja Turtles, and another one in this mythical world. Let's say that Easter bunnies can lay the colorful Easter eggs that we hunt on Easter. Transcription is where you're essentially turning DNA to mRNA. A key factor is the 3' A tail. The 3' A tail helps tell the RNA polymerase when to stop transcribing or termination of transcription. Since the 3' end is cleaved in transcript, but transcription can still continue. It also allows for the cleavage of 11 to 30 NTS downstream of consensus sequence. Pre-mRNA is what is cleaved at the 11 to 30 NTS downstream. Polyadenylation occurs next, which is where 50 to 250 adenine nucleotides are added to the 3' end by the enzyme poly-A polymerase. Translation is where you're taking that mRNA and translating it to RNA, which is how we get the amino acids to make the proteins our body needs. A key factor is once again the 3' A tail, which helps to initiate translation. It accomplishes this by aiding in the attachment of the ribosome to the mRNA. The 3' A tail also protects the mRNA during all of this. It protects it by the length of its tail because that tells us how stable it is. The tail will shorten as the mRNA ages. When the tail is too short, the mRNA is recycled. So let's say you have this sequence here and you want to transcript it and translate it. So first you find the start codon in the top strain, which is ATG universally, and then you also go until you find the start codon. So to transcribe that area, you're going to turn the T's to U's, which is how we get from DNA to mRNA, which I've done that here. For translation, you're going to take mRNA and turn it to RNA, which RNA eventually turns into the amino acids or the proteins. So I took this strand, and like I did up here, I blocked off the 3 letters that are going to make an amino acid. So when we translate it, we're going to have start, WIT, stop, because like I said, start, ATG is the universal for start, which start can also be represented by an M, which is also what I did. So now we have MWIT, stop. So let's say you want to learn about the mutations that can occur with that coding. So a silent mutation, which is seen here where I turn the T to a C, is where it codes for the same amino acid. And then you have a nonsense mutation, which I turn the G to an A, and it kind of changes the codon so it becomes a stop codon, which you can see that here. When I turned the G to an A, it went from a W to a stop. Then you also have a read-through mutation, which is where the stop coding is changed into a different coding that allows for the protein to be longer. So as you can see here, I changed the G to a T, which then changed that TGA, which was a stop codon, to a TTA, which became the L. And then the last one is a missense, which is where you change the amino acid and it may alter the overall function to make the protein functionable or make it non-functionable. So I changed that A to a C, which changed that T to a P and left everything else the same. But then you can also have transition or transversion, which transition is where you take a purine to a purine or pyrimidine to a pyrimidine. And then transversion is where you have a purine to pyrimidine or a pyrimidine to purine. So the two transversion mutations in this case are read-through and missense, and the two transitions are silent and nonsense. So let's say for missense, for example, one example would be if the Easter bunny can lay those Easter eggs like mentioned before, or if it prevents them to not lay those Easter eggs. Here are my resources, and thank you.